Bioremediation and biotechnology. Bioremediation is the process of using living organisms, mainly microorganisms, bacteria, fungi, algae, and sometimes plants, to clean up or detoxify polluted environments, such as soil, water, or air. It is a natural, eco-friendly, and cost-effective method to remove or neutralize contaminants like oil spills, heavy metals, pesticides, and industrial waste from the environment. Bioremediation depends on the activity of biological agents that can degrade, detoxify, or accumulate pollutants such as 1. Bacteria. Most important agents. Examples: Pseudomonas putida, degrades hydrocarbons, oil spills. Bacillus subtilis, breaks down organic waste. Alkanivirax orchimensis, marine oil degrading bacterium. Deinococcus radiodurans, resistant to radiation, used in radioactive waste cleanup. 2. Fungi. Decompose complex organic pollutants using enzymes like lactase and peroxidase. Examples: Phanerochaete chrysosporium, white rot fungus, degrades lignin, dyes, and pesticides. Aspergillus niger, removes heavy metals. 3. Algae. Absorb and accumulate heavy metals and nutrients from wastewater. Example: Chlorella vulgaris, Spirogyra. 4. Plants. Phytoremediation. Plants absorb or degrade pollutants through roots and leaves. Example: Brassica juncea, Indian mustard, accumulates heavy metals. Process of bioremediation. Bioremediation can occur naturally or be engineered for faster results. 1. Identification and assessment. Identify pollutants and the extent of contamination. Select suitable microorganisms or plants capable of degrading them. 2. Stimulation or introduction of microbes. Either stimulate native microbes by adding nutrients or oxygen, biostimulation, or introduce specialized microbes, bioaugmentation. 3. Degradation process. Microbes metabolize pollutants as their food source. Pollutants get converted into less harmful products, such as CO2, H2O, and biomass. 4. Monitoring and control. Regularly monitor pH, temperature, oxygen, and pollutant concentration until cleanup is complete. Types of bioremediation. Type. Description. Example: in situ bioremediation. Treatment occurs at the contaminated site itself. Bioventing, biosparging. Ex situ bioremediation. Contaminated material is removed and treated elsewhere. Composting, bioreactors. Phytoremediation. Using plants to remove or neutralize toxins. Brassica juncea, populous species. Micromediation. Using fungi to degrade contaminants. Phanerochaete chrysosporium. Bioaugmentation. Adding engineered or cultured microbes to speed up degradation. Pseudomonas putida addition in oil spills. Importance of bioremediation. Eco-friendly, uses natural organisms, minimizing chemical usage. Cost-effective, cheaper than physical or chemical cleanup methods. Safe and sustainable, converts harmful pollutants into harmless substances. Wide applicability, effective for oil spills, heavy metals, pesticides, and industrial effluents. Prevents ecosystem damage, restores soil fertility and water quality. Reduces waste, converts waste into usable biomass. Limitations of bioremediation. Works best under optimal environmental conditions, pH, temperature, oxygen. Some pollutants are non-biodegradable, example plastics, certain heavy metals. Slow process compared to physical or chemical methods. Difficult to apply to deep underground contamination. Role of biotechnology in bioremediation. Biotechnology plays a crucial role in enhancing, controlling, and optimizing the natural process of bioremediation. It uses modern biological tools and techniques to develop or modify organisms, mainly microbes and plants, that can degrade, detoxify, or absorb pollutants more efficiently. 1. Genetic engineering of microorganisms. Modern biotechnology allows scientists to genetically modify microbes to improve their bioremediation abilities. Examples: superbug Bionanda Chakrabarti, Pseudomonas putida. A genetically engineered bacterium capable of degrading multiple hydrocarbons from oil spills. Recombinant E. coli, engineered to degrade toxic compounds such as mercury or aromatic hydrocarbons. Modified Deinococcus radiodurans, can clean up radioactive and toxic heavy metals due to high radiation resistance. Purpose is to create designer microbes that can tolerate extreme conditions, break down complex or recalcitrant pollutants, target specific contaminants, oil, dyes, pesticides, etc. 2. Phytoremediation enhancement. Biotechnology improves plants' ability to remove toxins through genetic modification and symbiotic association with microbes. Biotech applications. Transgenic plants engineered to express metal-binding proteins, metallothioneins, Phytoskeletons, for heavy metal absorption. Use of rhizosphere engineering, introducing beneficial microbes near plant roots to boost pollutant degradation. Example, Brassica chunsi, Indian mustard, engineered to accumulate more cadmium and lead. 3. Enzyme technology. Biotechnology helps identify, isolate, and mass-produce specific enzymes that degrade pollutants. Examples: lacases and peroxidases, from fungi used for dye and pesticide degradation. Dehalogenases, oxygenases, and lipases, used for breaking down hydrocarbons and chlorinated compounds. Immobilized enzymes used in bioreactors for wastewater treatment. 4. Metagenomics and bioinformatics. Modern biotech tools like metagenomics, transcriptomics, and proteomics help scientists study microbial communities in polluted sites without culturing them. Benefits: identify novel pollutant-degrading genes and pathways. Discover new species with bioremediation potential. Design microbial consortia, mixtures of microbes, for specific contaminants. 5. Bioaugmentation and biostimulation. Bioaugmentation, using biotech-selected or genetically improved microbes to enhance degradation at contaminated sites. Biostimulation, adding nutrients or electron acceptors to stimulate native microbes. Biotechnology helps design nutrient formulations that optimize microbial activity. 6. Bioreactor design and process optimization. Biotechnology helps design controlled bioreactors, example slurry phase, membrane bioreactors, where conditions like pH, temperature, and oxygen are optimized. Enables large-scale cleanup of industrial effluents and oil-contaminated soils. 7. Biosensors for pollution monitoring. Biotechnology develops biosensors that use microbes, enzymes, or DNA probes to detect pollutants and monitor cleanup progress. Examples: bacterial biosensors for detecting BTEX compounds, benzene, toluene, etc. Enzyme-based biosensors for heavy metal or pesticide detection. Importance of biotechnology in bioremediation. Role. Description: improves efficiency. Engineered microbes degrade pollutants faster and more completely. Expands pollutant range. Enables cleanup of complex and synthetic compounds. Environmentally safe. Uses natural or modified organisms instead of harmful chemicals. Site-specific application. Tailor-made microbes for different contamination types. Cost-effective. Reduces expenses compared to physical or chemical cleanup methods. Limitations/challenges. Risk of gene transfer from genetically modified microbes to native species. Environmental safety and ethical concerns about GMOs. Regulatory hurdles and need for biosafety assessment. Some pollutants are still non-biodegradable, example plastics, PFAS. Thanks for watching.